All right, our next topic for Chapter 9, and again, we're some of this information is coming from Chapter 11 as well, where the generation of action potentials, nerve impulses, is discussed. So we're going to go ahead and pull in some of that information from Chapter 11 for Unit 4. So our next topic is going to be membrane polarization, and as I mentioned at the end of the last video lecture, membrane polarization is what is required to generate these electrical signals by motor neurons. And as we'll see also, not only do motor neurons generate these signals, but also other neurons within the nervous system. And also these signals wind up being generated on the sarcolemma, the plasma membrane, cell membrane of your muscle fibers as well. All right, so what do we mean by polarization? So polarization is telling you have, I guess I'm not really hyphenating that right, but whatever. Polarization is telling you that you have a difference from one place to another, kind of like the North Pole and the South Pole, or a better analogy for what we're covering here would be like a battery. You've got a positive pole and a negative pole on a battery. And that's what we're talking about with the cell membranes of motor neurons, and as we'll see also your muscle fibers. Those muscle membranes have to be polarized, which means you need an overall charge difference. You need a difference in total electrical charge on one side of the membrane versus another. And as we're going to see, in your extracellular fluids, you tend to have a greater concentration of positively charged ions versus the intracellular fluids. Now you have positive and negatively charged ions in both locations, but if you sum up all of the charges, all of the pluses and minuses, remember 1 plus and 1 minus would cancel each other out. That would neutralize those charges. So if you total up all the positively charged ions and the negatively charged ions in your ECF versus your ICF, you would find that overall you have more of a positive charge in your extracellular fluids versus the intracellular fluids, where you have a little bit more of a negative charge. It's not a huge difference, but it's enough that it leads to the ability to generate these electrical impulses within the nervous and muscular systems. All right, so what is the major basis for these differences? Your ECF, you might remember we talked a little bit about this when we went over body fluids at the end of Unit 2. The extracellular fluids have a higher concentration of sodium ions. So you have more sodiums out here than you do inside the cell. You do have sodium inside the cell as well, but you don't have, the concentration is not nearly as high as it is outside. On the other hand, your intracellular fluids tend to have a greater concentration of potassium ions, abbreviated with a K. All right, so I know both of those are positively charged. Potassium ions are not the only kind of ions that you have in your intracellular fluids. There are other negatively charged substances inside the cell, including a lot of proteins that have an overall negative charge. Some of the proteins are chains of amino acids. Well, some of those amino acids can have negative charges, and if you sum up all the charges within a protein, you can come out with some that have an overall negative charge to them. So because of that, if you total up if you look at the difference in charge in your ECF versus your ICF, the outside of the cell is going to be more positively charged than the inside. So that's kind of your take-home message there. And that's called membrane polarization, this difference in overall charge outside versus inside. The outside is like a positive pole, the inside is like a negative pole. All right, and if you could, that is something that can actually be measured, and values can be assigned to it. And I know what they're showing you here. Okay, so we've got a motor neuron down here on the bottom, and then we're blowing up a part of its axon. Remember, an axon is an appendage it's an extension of the neuron itself. And so, it's going to have a cell membrane covering its surface also. And this is a voltmeter. So the difference in charges between the inside and the outside can be measured in volts. That's voltage. And we're not going to get into the nitty-gritty details of what voltage is, but that is a measurement of the charge difference between the outside of the cell and the inside of the cell for our purposes. The difference is it has to be measured down in the millivolt range, 1/1000th of a volt, or MV is the abbreviation for that. And if you could actually take a voltmeter, a little microscopic voltmeter, and stick that down inside the fluids in the axon of a motor neuron, you would actually measure the voltage, the charge difference between the outside and the inside would be negative 70 millivolts on average, which just means the inside is more negative than the outside. That's the easy way to remember that. All right, this measurement, minus 70 millivolts, is called the resting membrane potential of a motor neuron. Resting membrane potential. Potential sounds kind of scary. Don't worry about it. The reason it's called a potential is because when you have this charge difference across a membrane, where you've got a greater positive charge on one side and a greater negative charge on the other, there is potential energy associated with that. Just like in a battery where you have a positive pole and a negative pole, there's potential energy that's associated with that, and you tap into that to power things that you want to power with a battery. So same thing with your cell membranes. When you have a charge difference like this, there is potential energy associated with that charge difference. So that's where the term potential comes from. Don't stress out too much over that terminology. All right, resting membrane potential. This is what you would expect to find on a typical motor neuron when that motor neuron is at rest. And being at rest means that it is not generating a nerve impulse. Okay, so it's not doing any signaling, it's resting. Okay, so again, let's see, I think I've probably covered most of this already on the diagram. Resting membrane potential, typically the value associated with that in a motor neuron is minus 70 millivolts. And how is that maintained? How is it generated? It's the difference in the ionic makeup, the differences in the concentrations of your positive and negative charges in the ICF, intracellular fluids, versus the ECF. And this winds up being maintained. How does this maintain? How do cells control this? Differences in permeability. So permeability means the ability of an ion to be able to cross the plasma membrane from one side to the other. So there are differences in permeability between different kinds of ions. Sodium and potassium are the ones we're mainly going to be focused in on here. And remember back in Unit 1, we talked a little bit about passive transport and active transport, how ions and other substances get across cell membranes through membrane proteins, and sometimes those are channels and sometimes they're carriers. Well, now we're going to be seeing some examples of these things at work. So ions, anything that's positively charged or negatively charged, cannot freely diffuse across a cell membrane. Things that are positively charged and negatively charged love water, so they're going to stay in the ICF or the ECF where they got lots of water molecules to get all warm and fuzzy and cozy with. So if ions are going to be able to move across a cell membrane from one place to another, you've got to have proteins in the membrane that act as channels to allow those ions to cross. That's the only way they can get across. So you literally open up like a little tunnel through the protein, and that allows those to flow across. All right, this diagram is showing you a little bit about how Well, how is this maintained? What are we talking about here? What's going on? There is a link here in the PowerPoint to an animation which shows how this is maintained, and as I mentioned on an earlier lecture clip, your textbook authors have generated some, or your textbook publishers have generated some excellent animations that really give you a nice visual on these things, so I encourage you to watch those. I plan to either embed those or link those directly out of Blackboard for you. All right, so what we're showing, in order to keep our resting membrane potential at this value, minus 70 millivolts, then we need to make sure we keep the sodium concentration higher outside the cell and the potassium concentration higher inside the cell. Those are the two key things. And again, I know both of those are positively charged, so that can seem a little bit counter to what we're talking about, the outside being more positive than the inside. And again, it's not that the outside is all positive and the inside is all negative. If you sum up all of the charges, outside versus inside, the outside is a little more positive than the inside is. The inside's a little bit more negative. So this minus 70 millivoltage really is based on the differences in sodium and potassium concentrations on either side of the membranes of these cells. All right, and so down here they're trying to give you an idea from the textbook about how this difference can be maintained. And so we'll just kind of walk through this here. So what they're showing you here, here's a cell membrane. And what they're saying here is, suppose you had a cell membrane, you got a motor neuron, and its membrane only had potassium channels in it. So these purple things you see there, those are potassium channel proteins, and they're embedded in the cell membrane. So they're opening up a little passageway, and that passageway is the perfect fit for potassium ions. It doesn't allow any other type of ion to pass across. That's something to keep in mind as well. Many of these channels only let one particular kind of ion move across. That's important. So what they're trying to show you here is if that was the case, it's not, but if that was the case, you have more potassium ions inside the cell. The concentration of those is greater inside the cell than outside. So which way are those potassium ions going to want to flow? If we think back to diffusion, they're going to want to go from where they're more concentrated to less. So they're going to go from inside the cell to outside. All right, so let's think about what would happen to your membrane voltage, that charge difference, if that happened. If you opened up channels to potassium and potassium flowed out, well, potassium ions are positively charged. So guess what? If you just let those flow out of the cell, the inside of the cell, the voltage would actually drop. It would become more negative. It would be about minus 90 millivolts. All right, and again, that's because you're letting more positive ions out, which is going to make the inside more negative than it would be otherwise, more negatively charged overall. Okay, and so now they're saying, all right, but now let's put some sodium channels in there. So now you've got membrane proteins that only let sodium pass through. Now we said sodium is more concentrated outside the cell than inside. So which way will the sodium ions want to flow? They're going to want to flow back inside the cell. That's going to be their natural way to want to diffuse. They can't do that unless they have a channel protein to pass through. All right, so if you had both potassium channels and sodium channels, allowing potassium to flow out and sodium to flow back in, that would actually bring the interior charge back up to about where we want to be, about minus 70 millivolts, because you're letting some more positive charges back in. So instead of being way back down there at minus 90, that's going to allow us to come back up to minus 70. All right, so just having those two types of channels, those are called leak channels, which they'll talk about in this animation when you watch that on Blackboard. Those help maintain this proper voltage. Now, if you just let just those leak channels themselves continue letting potassium out and sodium in, eventually the voltage is going to get screwed up. You just can't let things leak like that all the time without compensating for it. And so that's where you have another type of protein that's very important. It's called the sodium potassium pump. And that's what they're showing you over here. What that does is it pumps excess sodium out of the cell and excess potassium from outside back inside the cell. All right, so when you have too much potassium out here, it's going to pump that back in. When you have too much sodium in here, it's going to pump it back out. That's called the sodium potassium pump. You're pumping, so you're forcing those ions to go against the way they want to diffuse. Sodium does not want to flow back out of the cell because it's still more concentrated outside, so you've got to pump it out. You've got to force it out. Same thing with the potassium. Potassium doesn't want to naturally diffuse back into the cell, so you've got to pump it back in. You've got to force it back in. That requires ATP. ATP, remember we said ATP fuel molecules, those are combusted, those are broken down to provide energy to power things that require work, and that includes these pumps. That might sound kind of abstract, but as it turns out, these sodium potassium pumps are so critical that they actually use up a lot of the ATP that these cells make. Much of the ATP that your body produces is actually used to run those pumps and help you maintain this minus 70 millivolts of resting potential across your cell membranes. Believe it or not, that's where a lot of your ATP goes, so that's where a lot of the energy from the foods that you consume, that's where it goes. That's where a lot of the oxygen gas molecules you breathe in go towards helping make the ATP that's needed to run those pumps. Just a little bit of trivia there for you. All right, so now we've seen, okay, so what did we mean by cell membranes being polarized? And our goal here is to get to a nerve impulse. And another name for that is an action potential. And again, don't be scared by the word potential. That's just referring to the energy, this electrical energy that exists because of that charge difference across the membrane. So the subject of our next lecture will be, what do we mean by how do we get to a nerve impulse, this signal that's going to be sent down a motor neuron and toward a muscle fiber.